have you on Planetterrian Daily, episode 112. Today is July 6th, 2026. Some interesting findings to cover today. A blood test measuring ApoB particles may prevent more heart attacks than standard LDL cholesterol screening while staying cost-effective. Researchers compared ApoB particle counts with standard LDL measurements in large population datasets. ApoB directly quantifies the atherogenic particles that enter artery walls. The test remained cost-effective within U.S. healthcare spending thresholds while projecting fewer heart attacks and strokes. Current guidelines still rely primarily on LDL cholesterol values. Wider adoption would require updated laboratory reference ranges and clinician education. CSIRO researchers developed FlockMate, a 2-year project that uses sensors to capture live weight and fleece weight as sheep move freely through their environment. The system avoids labor-intensive manual measurements that stress animals and disrupt farm routines. It records data continuously rather than at discrete handling events. Farmers can track flock changes without repeated physical interventions. Next steps include field validation across different sheep breeds and pasture conditions. A new systematic scheme uses machine learning to classify structural changes in water across temperature and pressure ranges. Water expands on freezing and shows other anomalies tied to shifts in its hydrogen bond network. The model distinguishes ordered and disordered molecular arrangements without relying on predefined geometric templates. Validation against experimental scattering data confirmed the classifications. The framework can now be applied to other anomalous liquids. Zoologists analyzed attacks that occurred between 2020 and 2021 to identify conditions that increase conflict between people and urban coyotes. The study documented changes in coyote behavior linked to food availability and human activity patterns. Recommendations focus on securing garbage, supervising pets, and maintaining distance during known denning periods. The findings apply to other cities experiencing similar wildlife adaptation. Ongoing monitoring will track whether the measures reduce encounters. Lipid droplets rich in triacyl glycerols and cholesterol esters were shown to support precise formation of supramolecular polymers inside living cells. The neutral lipid environment modulates polymerization kinetics and polymer length. Researchers varied lipid composition to achieve on-demand control over assembly. This mechanism offers a route to regulate cellular functions through engineered polymers. Future experiments will test whether the approach can deliver therapeutic cargoes within droplets. Now, shifting to a very different area of research. Artificial lighting at night traps woodlice in continuous looping paths never previously recorded in natural settings. The behavior occurs when light disrupts the animal's orientation cues on the ground. Thousands of individuals can become caught in a single illuminated patch. The observation expands known effects of light pollution beyond flying insects. Reducing unnecessary outdoor lighting at night could limit these traps. While light alters small animal behavior at night, researchers have also engineered material shrinkage to produce functional curved surfaces inspired by living organisms. A programmed shrinkage technique mimics how flower petals and animal limbs form curved shapes suited to specific functions. The method creates surfaces that aid water drainage, improve strength, or enhance aerodynamic performance. Researchers tuned material properties to control curvature at multiple scales. The approach works with a range of base materials rather than requiring specialized molds. Further work will test load-bearing capacity and fluid flow benefits in engineered prototypes. A large biomonitoring study measured 45 environmental chemicals in maternal blood and associated several with earlier delivery and lower birth weight. Replacement compounds introduced after older chemicals were phased out showed similar associations. Exposure occurred through everyday products and diet. The findings highlight the need for broader chemical screening during pregnancy. Follow-up work will examine mixtures rather than single compounds. A registered report tested sequence-based predictors on a curated set of 3,814 human proteins with known subcellular locations. Performance varied widely across organelles and protein classes. The study provides standardized metrics that future algorithms can be measured against. Independent validation reduced overfitting concerns common in earlier, smaller datasets. Updated predictor versions will be reevaluated on the same benchmark. An analysis argues that current consent models overlook patients' ongoing control over their health data after initial collection. The authors outline how explicit data rights could clarify secondary use, sharing, and withdrawal. The framework aligns with existing privacy regulations while addressing gaps in research reuse. Implementation would require changes to electronic health record systems. Pilot programs at academic medical centers are under discussion. A perspective describes how near-term quantum devices paired with classical computers could solve specific molecular simulation problems before fault-tolerant quantum machines arrive. The approach targets protein folding and enzyme design tasks that remain difficult for conventional methods. Early demonstrations have focused on small molecules. Scaling to clinically relevant proteins remains the central engineering challenge. Funding agencies are soliciting proposals that combine both computing paradigms. Engineered control over physical form leads naturally to questions of evolutionary form. Analysis of genetic data resolved the long-standing Zoroptera problem by placing these tiny, termite-like insects within their correct evolutionary lineage. The group contains only a few dozen known species that live under bark or in decaying wood and look remarkably similar. Whole genome comparisons revealed relationships hidden by their conserved morphology. The study integrates fossil and molecular evidence to build a clearer insect tree of life. Additional sampling of understudied tropical populations is planned. Clarifying hidden relationships in nature brings us to newly identified shared vulnerabilities among gut bacteria. Researchers pinpointed enzymes used by E. coli, Shigella, and related species to penetrate the gut mucus layer. Blocking these enzymes prevented the bacteria from reaching the epithelial surface in animal models. The common target raises the possibility of a single vaccine covering multiple pathogens. The mechanism centers on mucus degradation rather than toxin production. Human safety studies of candidate inhibitors are the next planned stage. From microbial mechanisms inside the body, we turn to a special segment on how everyday lighting affects the smallest soil creatures. You've probably heard that artificial lights at night mainly confuse moths and other flying insects drawn to bulbs. In reality, the same lights can trap thousands of pill bugs, small ground-dwelling crustaceans, in endless circular paths. Right now, as streetlights switch on in neighborhoods worldwide, woodlice that rely on darkness for orientation lose their bearings and loop until exhaustion. Researchers documented these death spirals for the first time in natural settings, showing that light pollution reaches even animals that never leave the soil surface. A single illuminated patch can contain thousands of trapped individuals, illustrating how pervasive the effect has become. The next time you notice outdoor lighting left on overnight, remember that reducing unnecessary illumination protects more than just nocturnal flyers. It also prevents disorientation in the smallest soil creatures. Watch for city lighting ordinances that incorporate invertebrate behavior data alongside energy savings. The spacecraft completed a 1 billion kilometer trajectory to collect the first asteroid samples for return to Earth. Onboard instruments will characterize surface composition before sampling. The mission timeline spans more than a year of cruise and proximity operations. Data from this encounter will inform models of asteroid formation. Sample return is scheduled for subsequent mission phases. A chirping radio signal from a distant supernova remnant provided direct evidence that a newly formed magnetar can power the explosion. The detection used general relativity to model the dynamics of the collapsing core. This marks the initial confirmation that such extreme magnetic fields arise at the moment of neutron star formation. Continued monitoring will track how the magnetar's spin and field evolve. The finding refines theoretical predictions for the brightest stellar explosions. Next time, we'll be watching for continued field validation of the flock mate sensor system across different sheep breeds. That covers today's science and health news. Share this with someone who's curious about the latest research. I'm Patrick in Vancouver. See you next time. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. And before you go, this show is part of the NARA Network, a family of daily podcasts on tech, science, and markets. Each one a short, sharp briefing you can finish on the commute. If today earned its place in your feed, here's your next listen. Modern investing techniques, modern strategies for Canadian and US investors. Find every show free at naranetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.